Am I audible? Okay. Yeah. So we'll wait for a couple of minutes and then we'll begin. If somebody more wants to join, then we'll wait for a few more minutes and then we'll start. Hello everyone. So is my slide visible to you all?
Yes, sir.
Can you see the slide? Okay, fine.
Yes, sir.
Thanks. Uh, so hope I have waited for the enough time for the people to joining so that I should not go again to admit the people. Okay, so yeah, good evening everyone and welcome to the week second of the NPTEL course of Techniques of Material Characterizations of the Doubt and Problem Solving session. Myself, Saurabh Soni, research scholar from IIT Bombay, from the Department of Metallurgical Engineering and Material Science. And since the last class was our first class, and we went very slowly, and we discussed a lot of things from the very basics, since the many people are from the different background. And for me it is very important, to take you all from the same page, so that you all would be able to do your assignments in an appropriate manner. At least I would expect those who are not from core background, at least they can also solve more than 50%. So that is my more main motivation behind taking this doubt and problem solving sessions. So hope the last class would be somewhat useful for you when while solving your assignments problem and while watching your classes. So as I said in the last class, to watch at least more than 3 lectures. So how many of you have watched the lectures of week 2? I just wanted to I just want to know. Or how many lectures have you watched? So Nivedita is saying 3 lectures. Salis is 3. Nishant has watched all the lectures. I want I want to hear from everyone. Arun is saying 4, 3. Don't be shy, even if you have not watched. I mean, you can just openly say. I just want to know that who is putting how much effort. Not started week 2. Okay, this is my first lecture. Okay. So Sakshi, this is your first lecture. So in the last lectures, I said that at least before coming to the class, watch at least 3 lectures, so that you will feel, um, relatable, whatever I will be explaining. Otherwise, it will be like, out of the box. And if you have not studied before, then it would be very difficult, because here I cannot take each and every concepts. Here I will be taking only few concepts which can be helpful for your assignments, not for for all your lectures. So it is always advisable to at least watch 3 lectures before coming to the class, so that you will not feel that I am explaining something randomly. And yeah, it would be useful for you. What about others? What about others? Only this many people have watched. What about rest? Rest for enjoying. Okay, no issue. It would be always benefited for you if you will watch. Otherwise, uh, just just a minute. Okay, yeah, fine. So let's begin our today's session. Sorry, uh, I have forgot to change. Okay, I'll do it later. Okay, so let's begin with our first problem. And here the question is in your screen. In which type of optical microscope? So in the last class I have explained about microscopy, different kind of microscopy in a brief, and also I have said that what kind of, where can we use this different kind of microscopy, such as scanning electron microscopy, optical microscopy. And also we have dealt with a little bit about the optics, how this optical and the how these lenses are arranged in what manner. And we also saw some problems. So here it is asking about which type of optical microscopy. So now you see that in the first week we were just getting the basics. Now we are getting deeper into the optical microscopy, right? So here, from here, our microscopy is started in a detailed manner. So here it is asking both the light source and the objective lens are present in the same side of the specimen. Okay. So there are a different kind of a microscopy. You must have watched the lecture, so you would be knowing this. The reflected light microscopy, transmitted light microscopy, both reflected and transmitted light microscopy, and then none of this. So can you tell me that if you have seen the lectures, that's what I was saying, and the participation is very very important in this class because I want to hear each and every one from you so that I can make it more productive and we can have a more discussion on that. So this is very basics and simple question. So what could be the answer where the light source and the objective lens are present in the same side, not in the opposite sides? So okay, um, reflected light B. Someone is saying B. Someone is saying A. Reflected. So most of the people are saying A. Reflected. Okay, just a second. Okay, so people are guessing that it is reflected light microscopy. Okay, so let's move and we'll see what could be the answer of this. Okay, yeah. So this is reflected light microscopy. Okay, so if you have seen in the lecture, this is the arrangement how the lenses are arranged in the reflected light microscopy. So as from the name itself, it is clear reflected. Reflected means what? Where the light is reflecting, and transmitted. Transmitted means what? Where the light would be transmitted. So here the reflected light microscopy is often referred to as incident light, epi-illumination or metallurgical microscopy. So we also know this is a metallurgical microscopy because it is used more oftenly in the for the metallurgical sample. The optical pathway. Now here it is explaining about the how the light is going and what kind of a path it is taking. For the reflected light begins with the illuminating rays originating in the lamp housing for reflected light. Okay, so here is your lamp filament. So from the lamp filament your light will be generating. This light next passes through the collector lens. So from here the light will generate and it will pass through the collector lens. Okay, so in the last class I have told about that how this the rays are when they pass through the lenses and how the path of these lenses sometimes it converges, sometimes it diverges, and it basically changes the path. Okay, it depends on the type of the lenses basically. So and into the vertical illuminator. So where is the vertical illuminator? Here. Okay, so here this is your this is filament image at condenser aperture diaphragm. Okay, and here again one lens will be there, and this is field diaphragm, control field diameter, and this is your field lens. So after passing through the vertical illuminator, the light is then reflected. Okay, so here after this the light is, what is happening, the light is then reflected by a beam splitter. So here the light is coming in the particular direction, and here you will be having beam splitter, or you can say the mirror. So basically because of this it is known as a reflected, because when some light is coming in the mirror, then what will happen, that will reflect, that cannot transmit from the mirror. That's why we call it reflected. Reflected means someone is coming here and it will get reflected, it cannot be passed through this mirror. Okay, now after this throw the objective to illuminate the sample, and now after what will happen here, when it will reflect from the mirror, it will through the objective. This is your objective lens, and through this it will fall onto the specimen, or you can say it will illuminate the specimen. Now the light reflected from the surface of the specimen, okay, re-enters the objective and passes into the binocular head, where it is directed either to the eyepiece or to a port of photomicrography. So now here this is telling about this. Here your image plane and here your image will form. Okay. So this is basically a brief about the reflected light microscopy. Now let's see the transmitted. So this is, if you have been to any laboratory or if you have seen the transmitted light microscopy, it basically looks like this. This is the optics. I mean, this shows that how the principle and how basically the lenses are arranged where we keep our samples. But outside, from the outside, if you will watch, you can't see this kind of arrangement. This is different. It is a somewhat, simplest way to explain that what is happening, but in a reality it is not like that. So in the reality, when you will see any kind of instrument, it is different, but it is a simple way to explain the concept. So hope if you have been to any kind of a laboratory, if you have seen the transmitted light microscopy or any microscopy, sometimes even if you will go for the checkup and all, they also have some kind of a microscopy. There you can see, our samples, blood samples and all. So they are also one kind of arrangement of the lenses. So yeah, this is transmitted. So the transmitted light microscopy is associated with the light passing from the source to the opposite side of the lens. Okay, so here what is saying? Light, so it is saying that light passing from the source, this is your source illumination, to the opposite side of the lens. This side is your opposite side of your lens. Okay, so here why it is saying, because the specimen is not at the end, specimen is at the center. That's why here when the light is coming, it is not reflecting, it is transmitting. Okay, there is a difference. Transmitting means it is going through that sample, and reflecting means it is not going through the sample, it is going and reflecting from the surface of that mirror or whatever you will say, but here it is passing through the sample. So this method is used to distinguish the morphological characteristics and optical properties of the observed material. Now, the light rays are organized and conducted through the instrument, so the light can be polarized and redirected. The light from the condenser will fill the plane of the objective and a ray of light will be projected to the, so it is basically saying about how the light is going and how it is passing through the different kind of lenses. So this is your, condenser aperture, and here you will have a condenser lens, and this is your specimen, and this is your immersion oil. Here will be your oil, and this is also immersion oil, and this is your objective rear focal plane, and at last you will be having this. So this is a sample is in between, it is not at the last. So the light passes through the sample. So here it is, just now I have explained, passes through the sample and it will go to the objective where the image will be magnified. Okay, so this is your objective and here your image will be magnified and you can see the zoomed version. So now what was our question? We have seen the enough of the explaining. Now the light source and the objective lens are present in the same side. So here this is your light source. Okay, and it is here, when you will see in this, here your objective is different at the opposite side and your light source is different side, but here it is not like that, it is at the same side. Okay, so that's why the answer is your reflected light microscopy. Okay, so the answer is reflected light microscope. So many of you have guessed it correctly. So yes, answer is reflected. So I hope what I have explained it is clear.
Yes, sir.
Yes or no?
Yes, sir.
So Nishant is asking how to analyze an opaque material with polarized microscopy or with upright my. So Nishant will come. Right now I am asking about this reflected light and the transmitted. What I have explained about this. Is it clear? We'll come to that point when we'll have further questions. Okay, yeah. Okay. So the next question is our contrast generation in optical microscopy is increased by what? So here it is asking about if you have seen the lecture, there is a concept of how what are the factors that affects the contrast and all. So here the options are your background subtraction algorithm using stain or dye in biological specimen, both by background subtraction algorithm and using dye in biological specimen, mounting of a specimen. So can somebody tell me that what would be the answer for this? Okay, so let me see what people have guessed. Uh, A, C, C, A, both C. So some are saying A, some are saying C. Okay. Can you tell me that what is contrast or how you measure the contrast? What is contrast? We know contrast. We sometimes see we click our photographs and we give this kind of interest statement that the contrast is good or the contrast is not good. Can you tell me that what is exactly contrast? How you will define the contrast? You should know the technical term that what is contrast and how you define the contrast.
Or maybe it is color difference.
I'll be reading your comments. You just tell whatever you feel. You often, call it that the contrast is not good of this image. What does it mean? How you will define the contrast? Color difference. Okay. Ability to distinguish color. Okay. Can somebody tell me in a more appropriate manner? I mean, I'm just expecting something that, but you people are not responding.
Hello, sir. Am I audible? Hello.
Okay, yeah. So Sarita is saying difference in color that makes object different from the background. So yeah, this is somewhat more technical in terms. Definitely, it is the distinguish between a different feature or the different color. But what I expected was the contrast is in any kind of a microscopy. It what it refers it is the difference in the brightness or the color between an object of interest. Object of interest means what we want to focus and its surrounding background. Okay, so with respect to its surrounding background, so it is a critical aspect that allows the visualization of details and structure. Okay, whatever the specimen you are measuring. Okay, so if higher will be the contrast, the visibility of the specimen's feature, making it easier to observe and analyze. Okay, so if we will say the contrast is higher, it will make your life easier in analyzing your sample because you can see the features more clearly. Somebody was saying here ability to distinguish different features clearly. So you will be going to distinguish between the different features. But how? The question is how. What is making it different? So the different is what? The color between an object of interest and its surrounding background. Okay, so that is something that should be distinguished. So then only we can say the contrast is good. So if you have seen the lectures, there are a different kind of a contrast. You can see a bright field contrast, phase contrast, differential interference contrast, dark field contrast, fluorescence contrast, polarized light contrast. So these are all some details because it is a very generic term. But here, when you will be as a material scientist, what kind of a specimen, if you have a biological samples, if you have a metallurgical samples, if you have a polymeric sample, if you have a metallic sample, ceramic samples, fiber samples, different kind of a sample. So in that case, you should have the knowledge that which kind of a contrast or the microscope is giving a what kind of a contrast that would be more suitable for your specimen, right? So yeah, so let's come to the question again. So now the it is this slide is asking about the enhancing the contrast in optical microscope. Okay, so here in transmitted light microscopy, the specimen quality does not always lend itself to easy observation and image recording with excellent contrast as in simple bright field imaging mode. So here the investigations dealing with inherently low contrast specimen, stained bacteria, tissue slices, some live cells rely on specialized contrast enhancing techniques for imaging of virtually transparent samples. This is what something I was saying that here different kind of a samples is there. So the contrast is very important. That what kind of a specimen with what kind of a specimen you are dealing. Sometimes biological samples like cells, tissues, bacteria, or the virus, it could be anything. In the course of examining unstained specimen, poor light absorption by the specimen results in extremely small variation in the intensity distribution difference between the specimen and the background. Okay, so it is asking if you will have a unstained, if you will not use any kind of a dye or this stain in the specimen, the light absorption will be poor. If the light absorption will be poor, how you would be able to distinguish between the specimen and with the background. So when the background is bright, okay, human eye requires local intensity fluctuation of at least 10 to 20% to be able to recognize the specimen details. So this is some facts which is measured or analyzed, but this level of modulation is seldom seen in with different with transparent specimen which are usually rendered almost invisible against background of similar intensity. So how you will be measure the contrast, it is the intensity of the specimen. S is representing specimen, B is what, background, okay, divided by background. So the this is specimen minus background divided by intensity of background into 100, because it is asking in the percent. So if somebody this kind of a problem or the numerical question would be coming, if they could give you the contrast of any image is this much percent and the background intensity is this, then you could be able to calculate that what is the intensity of your specimen. Okay, so in this formula they can ask any kind of a question, so please be remember this formula. So now here this is about the image intensity. Okay, so when the intensity of the image is increasing, okay, so this is the contrast and this is the background intensity effect on the contrast. Okay, so when this is about the intensity is increasing here, it is the intensity of the background. So when the background intensity here, the background intensity is zero point 0.5, zero point 0.1, so it is decreasing. So when it is decreasing, the contrast is increasing. Okay, so here it is asking about the contrast and optical microscopy is increased. Contrast will increase by what? Background subtraction algorithm. So when the background, sorry, when the background subtraction here, just a second. So when the background subtraction here it is increasing, background subtraction means the background intensity is 0.5, zero point 0.1, 0.01. It is decreasing. So your contrast is increasing. Okay, and also here it is what it is saying, when you will not be using any kind of a stain or the dye, then the poor light absorption will be there and you cannot be able to distinguish between your specimen and the background, then the contrast would not be your good. So you will require both the dye or the stain or the background subtraction algorithm. Okay, is it clear? Okay, is it clear what I explained just now about the contrast? Okay, yeah. So now I have one image. This you can see, this is the bright field illumination sample. Contrast comes from absorbance of light in the samples, and this is a dark field. Okay, so this is the dark field illumination, this is bright field. So here the contrast comes from the light scattered by the sample. So here you can see when you will give a bright field illumination, your sample will, the contrast is something like this, and this is, so the dark field and bright field will also again depend on what kind of a specimen or the sample you will have. In most of the cases, or or in some cases, we have observed that the dark field, this illumination gives a better contrast as compared to the bright field. But it again depends on the sample to sample and what conditions. So this is the image of how actually it looks. So here this is your background and this is your sample. So now the intensity is different. That's why you can be able to, you know, say that this is forming a contrast. So the answer was both by background subtraction algorithm and using dye in biological samples. Now, again, the next question is from the uh, contrast enhancement is more sensitive to what? Reflection mode, transmission mode, both reflected and transmitted mode? None of this. So, more sensitive. Can you tell me if you have seen the lectures? I would not explain this about this, because if you have seen the lectures of this contrast, then you can guess or you can give the answer. Where it is more sensitive, contrast enhancement, in reflection or transmission, or in both. Transmission, transmission, transmission. Can you tell me that why it is more sensitive in transmission? Or are you just randomly guessing? Can anybody tell me that why the transmission mode? Maybe we have biological system more in transmission. Okay, but why it is? I mean, more sensitive. That is fine. We can have any kind of a specimen. Depends on you. But why it is more sensitive? Light may pass through transparent samples. Is it? Is it necessary that your sample should be the transparent? So I will discuss this, and I want to ask of this. I want answer for this, and please be logical. I'll again ask this question in the next class, and definitely the answer is transmission, as you said, you all. But I want to know more about this. That why it is so. So please come with the answers. It is very simple, and maybe if I don't know whether you have watched or not. So please see the lectures and come with the answers. Okay, so the next question I'll move, and the optical tricks or the optical arrangement are only used to amplify contrast difference in dark field and polarized light microscopy, dark field and bright field microscopy, phase contrast and differential interference contrast microscopy, bright field, and the polarized light microscopy. So where you use only used to amplify the contrast difference. Have you have you seen a different kind of this, microscopy? Polarized, phase contrast. I think one of the student was asking about this few minutes ago. So yeah, please tell me where we only use to amplify the contrast difference. So make it very interactive. I mean, don't feel shy, or it's not somewhere that you will be judged, or I'm not giving any kind of a marks that and it is very simple. You just come with the some lectures, and it's just about the discussion. I don't want this to be one sided discussion. Just I'm giving the problem, I'm showing you the questions, and I'm only the discussing about the concept, and then I'm again showing the answers. So the questions, discussion, and answers. I just want a mutual participations. It would be a very helpful for me and helpful for you. You will also feel interest, and then you can before coming to the class you will have interest to watch the lectures, because here I'm making a atmosphere so that we can have a discussion. I don't want it to be a more like a class where I'll come, I'll teach, and I'll go. Otherwise you will lose interest in this course. And this is very very beautiful course, I'm again saying you, because irrespective of the branch, irrespective of the branch, I mean, this is something, characterization is useful for everywhere, whether you are from the physics background, you are from chemistry background, from the chemical engineering, from the mechanical engineering. In somewhere, if you will be if you will be having your job in the R&D section, you should know the characterization. Okay, and these are some kind of a skills or theoretical knowledge that will make you know a better understanding with the instrument. So that's what I'm saying. Take interest of this course. If you have taken this course already, then come with the interest and participate in the interaction. So yeah, just tell me which will be used to amplify the contrast difference. So Nishant is saying both bright and dark. Devika is saying show the answer, and I just show the questions. Phase contrast, and DIC, bright and dark. Okay, so someone is saying bright and dark, someone is saying phase and DIC, bright and dark. So mostly saying bright and dark, and someone is saying phase contrast and DIC. So I think conflict between B and C. So we'll see. Okay, so let's see. So this is a phase contrast technique, and here it used the optical tricks to translate phase shifts into grayscale amplitude values. Okay, so it is amplituding the value. All of the wave fronts are ultimately recombined to form the intermediate image by the objective lens. So here it is your light source. Okay, here your light will go, and this is your condenser, and here is your specimen plane, and this is your objective, and from this specimen your light will pass through it, and this is again go through the objective lens, and this is your face plate. Okay, from here if you have seen the lecture, then you would be knowing that what is the use of this face plate, and from here again your light will go, and this is your image plane where your image will form. So the wavefront that have all been retarded to varying degrees by details in the specimen are superimposed in the intermediate image plane, where this amplify each other, forming the final phase contrast image. Okay, so what is the answer? It is mainly about the phase contrast microscopy. Okay, so in the phase contrast microscopy, you will be amplituding your what? Contrast difference by using your optical arrangement. Okay, now the question is again about the contrast. Maybe I have few more question on this. Which of these provides better contrast? Just I think few minutes ago I have shown you the one images, dark field, bright field, both, or oblique illumination. So it is very simple. Just give it a frequent answers.
So I have a question. Hello.
Hello.
Based on that contrast enhancing techniques,
Uh-huh.
when you see the lecture, when you go to the page number 12, they, there it has mentioned that methods that enhance contrast include DIC, polarized light, phase contrast, and dark field microscopy. What do you mean by that then?
This one.
Yeah, yeah.
Okay, so this is about the how you will be amplifying the contrast difference. So you are not, I mean, you are asking about the question or about the microscopy you are asking?
Uh, Methods that enhance contrast.
Okay. So what is
That
your
is exactly different from the tricks, optical tricks, what you were asking.
optical tricks? It means, see, in every kind of a microscopy, you will be having a different kind of arrangement. Okay, the optical in like in the phase contrast, you will be using some, that will create a phase. Or if you will go to the dark field microscopy, your optical tricks, or in a better word if I will say the optical arrangement would be different. That's why because of this different kind of arrangement we known this as a different microscopy. Okay, in dic if you have seen the lectures, the dic, the optical arrangement of the lenses are different.
So ultimately, it is amplifying contrast, right?
Yeah, so there could be something which is amplifying the contrast. Amplifying the contrast, it means to say you will be able to distinguish more easily by using that kind of a microscopy. There are, in the material science world, there are few samples which are, a kind of very critical to analyze, because they don't have a much contrast. So you require some kind of a microscopy that has the potential to differentiate between the different components. Suppose if you will be seeing a cells, in the cells there are a different, if you know about the cells, in the cells you will be having a different things. So how you will be able to distinguish in that biological tissues or that cells that okay, this is your this part, this is your mitochondria, this is your I do not know about much about the biological samples. So if you will be knowing about this, then it is very difficult to analyze this kind of a sample. So there you should be having a something which can amplify your contrast. In the case of metallurgical sample, suppose if you are just watching some very randomly grains and all, there you, I don't think so that you will be require a more specific kind of a contrast microscopy. I think bright and dark is most widely used in the field of metallurgy and material science. So this is all about that. So if you want for some specific application, you can go for different microscope. Otherwise, what I have seen is the bright and the dark field microscopy. Only from there you can be, able to distinguish more easily of whatever the required samples.
So all these methods, like DIC, polarized light, all these enhance contrast, right?
Definitely. I mean, it's not about the enhancing the contrast. Definitely, it will enhance, but it depends again on the application. For what purpose you
Okay.
want to use a which? I mean, this is something very basically just like I have explained in the last class. You can't go if your sample you want to see something in a range, or let's say 100 micrometer, I think so, or the 500 micrometer, I think so your optical microscopy would be much more sufficient. You don't need to go for the SEM or the TEM image. If you want to go for the to see in the nanoscale range or in the atomic range, then only you would be, it is advisable to go for the TEM. You know what I'm you mean, what I mean about this.
Yeah, yeah, yeah.
So
So what is the answer for previous question?
so the
It's
answer
just
is DIC and the phase contrast.
okay.
Yes.
Thanks.
Okay. So just see the lectures one more time. If you will be having any doubt, then we can discuss further. And yeah, this is about. So this is very simple. Like again, I have just shown you this image few slides back, which provides a better contrast. Dark or field, dark or bright, or both. So one is saying dark field, another one is saying A. So what is A? A is dark field. Okay. What about others? Dark. So now I think you can say it much more clearly. You can see this is dark field, the right hand side. This is your dark field and this is your light field. So which one is better? Okay, so many of you are saying the dark, which is right. So here the dark field microscopy gives the better contrast as compared to the bright field. So it is again about the phase contrast microscopy. Can you tell me that in the phase contrast microscopy is popular for what? Opaque specimen, thin, transparent, and stained biological specimen, thin, transparent, and unstained biological specimen, or all kind of a biological specimen. So for which samples we can, use this microscopy? It is more about, I think, application oriented sample. It would be good because in the future you can deal with this kind of a specimen. Then it is very simple for you that in which kind of a microscopy you can use. So what is the answer? So someone is saying C, thin, transparent, and unstained. C, thin, transparent, unstained, and Arun is saying B. So most of the people are saying C. Okay, so let's check. Okay, so this is a phase contrast imaging of transparent thin specimen. So this is your cell, and this is the nucleus, and this is your cover slip. On top of this, you will, your cell is there, and this is your transmitted light illumination. So yeah, so very small difference exists between the refractive indices of the cells and their surrounding. Okay, so I think it is very basic about what is refractive index and all. So it is saying that when there is a very small difference, or you can say a very there is not much difference between the refractive index of the cells and the surrounding aqueous solutions, and within the cells between the cytoplasms and the cell nucleus. Okay, so this is your cell nucleus, and this is surrounding is your cytoplasm. This is cells. So no significant refractive index difference occurs in the cover slip and the surrounding medium. So in this case, the phase contrast makes this tiny difference in refractive indices. Okay, so this is again where your refractive indices between your specimen and the, you can say, whatever the solvent, or you can say the aqueous solution you are using, it is, if it is very very tiny or very small, then you can use a phase contrast microscopy. So here, by the use of optical manipulation, for example, it translates them into difference in intensity that can be visually observed and recorded. Okay, so here again, the optical effect employed in this case consists of a shift of phase relation between the light wavefronts traveling through different portion of the specimen. Okay, so here, when this is your like a phase, okay, when your light is traveling from here, the phase will shift. Okay, so how much? This is your delta 1. So again, this will come to this. Now when it will come to this place, okay, here you will be having a nucleus. Okay, this is your surrounding of your cells, not surrounding of the cells, surrounding of the nucleus. So here your phase difference will be different, so this will be shifting towards this side. So now you will be having a different phase shift. Why? Because you will be your light is passing through the different medium of a refractive index. Here the nucleus refractive indices is different than the cells, and this is where your nothing is present. I mean, there is no cells, no nucleus. Okay, so the higher the refractive index of a medium, okay, when the refractive index of your medium is higher, the smaller the speed of the velocity of the light in the medium. So during their journey through cell nuclei, cytoplasm, or water, the light waves are shifted. Okay, so here this is the light waves are shifted or retarded by small degrees, since this media have slightly different refractive indices. So that's why, just because of different refractive indexes, your light waves will shift. So this is your shift delta 1. Here this is your nucleus, so this is again a different light waves. I mean, the light waves will shift in a different amount. So this is all about this. So here, this is an example of the phase contrast microscopy of the living cells in bright field and the phase contrast. So this is your bright field. Again, like just few minutes ago, I was saying that again, every microscopy is having their own application. You can't deal your biological samples with a bright field microscopy. So in that case, you have to go for the phase contrast. Just now I was explaining that where you should go for the STM or the TM or the optical microscopy. In a similar manner, every microscope or the every instrument is having some limitations. That's why there is a different instrument. Because if you want to measure one property of instrument, and if some instrument is not able to fulfill that requirement, then there is a different instrument that would be a more beneficial or the more advisable for the those kind of a measurement. So here it is not useful if you will suppose you will have some biological samples, you want to see the cells and all. So if you will be using a bright field microscopy, you will be getting this kind of image. What kind of a measurement or the analysis you will be able to observe from this image? Nothing. So it is always advisable to go, sorry, it is always advisable to have some basic understanding, so that you know at least that what kind of a microscopy you should use. So here you can see how clearly your cells and all the features are visible as compared to bright field. So that's why we use this kind of a sample in the phase contrast microscopy. So yes, thin, transparent and unstained biological samples we use for the phase contrast microscopy. So now I think it would be a much more better or the clear as compared to what I was trying to explain since last from the beginning of this class, that why we go for a different kind of a microscopy. Is it clear somewhat? Yes or no? Okay, fine. Um Okay, so I'll give you the next question about this, and I'll be taking a 5 minutes break so that I can relax my throat. So meanwhile, you can also type your answers for this or give that why is it so if you have seen the lectures, or if you want to drink water and all, you can go. Okay, yeah. So, what is the answer for this question? C, phase contrast microscopy. So, three are saying C. C. What about others? Say something. At least you give answer to this simple question. Few people are not giving answer any. What about Yashika? Can you tell me what could be the answer? Sarita, I can't see your response. Priestley, Afzal, are you people on the are in the class or just joined the class and left?
To answer.
Okay, yeah. So this is very basic, simple question, and I think you all are most of you all are right. So this is a halo and a shade of artifacts in phase contrast optical microscopy. So here you can see a uh, halo kind of a shape. So this is a phase contrast is microscopy about that. So excellent method to increase contrast when viewing or imaging living cells. Okay, so again, like in the last slide, I have shown you that in the living cells we can we used to see the living cells or a biological samples for the phase contrast microscopy, but typically results in halo surrounding the outline of the edge's future. Okay, so this is your edge and this is your interest, and here you can see something kind of a surrounding you can see a hole or some this is some bright kind of a things you can see. Okay, so this is that effect shadow, or you can say a halo effect. So in the halos arises because of the configurational parameters of the phase contrast microscopy. Okay, so this is because of the configurational parameters. So the circular phase retarding ring located in the objective phase plate also transmits a small degree of reflected light from the specimen. So the presence and the severity of halos in phase contrast is depend on the refractive index of the difference between the specimen and so again this is because of the suppose here you will be having again in the this image you will be having a refractive index of this medium is different than the refractive index of your this place. Okay, so because of this there is a formation of your this ring kind of a structure surrounding to your surrounding your specimen or the cells which you want to focus. So here the area is having a lower refractive index. So suppose if one part is having a low refractive index, then the surrounding medium exhibit dark halos as opposed to the bright halos that can be observed surrounding high refractive index. So again, it depends on the higher lower index and the lower refractive index. So on the basis of that there is a formation of your so here in some places you can see a dark halos and in some places you can see a bright halo. So again it depends on the refractive index of your surrounding medium and your specimen, or you can say your cells. So I hope so yeah, most of you have guessed it correctly. Phase contrast optical microscopy gives this halo or the shade of artifacts. So yes, now we came to the differential interference contrast microscopy. So it is used for what? Like we have seen about the phase contrast. Can you see that why DIC is used for what kind of a specimen, or the thin specimen with reflected mode, thin specimen with transmitted mode, thick specimen with transmission mode. So what could be the right answer for this? So only one is giving answer C. What about others? Okay, so I will give two questions simultaneously. This is also one question. Wollstonecraft prism is made by two prisms fused each other having similar refractive index, different refractive index, similar reflectivity, different reflectivity. Have you seen the lectures of this where we are using this Wollstonecraft prism in which kind of a microscopy and how it is made when we use two prism, or when the two prism fused, then what could be the quality? Not watched. Nishant is saying B. No, at least. Okay. So if you have not watched, then yes. Um, when you will be seeing this DIC lecture, then here you can, there is a slide of requirement for DIC. So DIC microscopy can be utilized on virtually any upright or inverted microscopy as long as polarizer, condenser, and the prism can be installed. So here you can see this is your analyzer, here is your objective, and this is your prism, and this is your nose piece with prism, objective specific prism, and this is your polarizer, and here will be your condenser. So here you can see that Wollstonecraft prisms are a type of prism made of two layers of a crystalline substance such as quartz which due to the variation of refractive index depending on the polarization of the light splits the light according to its polarization. Okay, so here you will be using a Wollaston prism where you will be using a two layer of a crystalline, okay, such as quartz. So what is the main difference? Main difference is your variation in refractive index. Why there is a variation? Because here we want to create a polarization of the light, splits the light according to its polarization. So here the Nomansky prism, here also this is Wollaston prism, this is Nomansky prism, causes the two rays to come to a focal point. Okay, here this is two rays are coming, so if you want to come let it come to the focal point outside the body of the prism, and so allows greater flexibility when setting up the microscope as the prism can be actively focused. Okay, so here the question was about the Wollaston prism, so it is about the different refractive index we use in the two prism of the two prism which is fused in the Wollaston prism. So the answer is here different refractive index. Okay, so here this is also one slide which I could gather from the Wikipedia. Here you can go. So I would also suggest you people, if sometimes when I show you some slides or some lecture, even though if you are listening lectures and if you could not get it properly, you can try searching from your side. Nowadays there is a lot of Google and all. There are different kind of sources where you can go and just check that is it so, and if you are having any kind of a doubt, then you can go and read. So this is simple Wikipedia. So the Wollaston, it is about the Wollaston prism. So here you can see this is your Wollaston prism. So this is one part and this is two part. So both are having a different refractive index. So here you can see this is also Wollaston prism. This is one part. This is second part. So optical devices. So this is about that, and it separates the light into two separate linearly polarized outgoing beam with the orthogonal polarization. So here you can see this is your unpolarized light, and here this will split your light into two separate parts, and then it will again propagate and it will go through the condenser lens, objective lens, and again there is a Wollaston prism which will again combine these two lights, and here this is your unpolarized, and at last you will get what? Polarized light. So this is how, I mean, if you could not able to gather from the lecture, it is always advisable to go and search some more content on your topic. So I hope you can get some basic idea, and you can able to answer that what is Wollaston prism, what is, why it is used in the DIC. Now, which mode of optical microscopy gives depth perception? Yeah. So again, it depends on the application. Suppose if you will have some samples where you want to see some depth configuration, and it's not something that you only want to see the surface morphology. You want to see the jet along the jet direction also. So the polarized microscopy, DIC, phase contrast, or the DIC and polarized, which could be the option for this question? Can you tell me? Yes. So DIC. Okay. What about others? No opinion, no answer. Yeah. So the DIC is correct. So here it is asking about the depth perception. Okay. So here can you see in this image that there you can find some depth. Here you can see this is at some height and this is at the bottom. So it is possible in the DIC mode. So image is generated from two identical bright field images being overlaid slightly offset from each other, and the subsequent interference due to phase difference converting changes in phase to a visible change in darkness. Okay. So here you can see this is, you can see the height. If you will have a z axis or if you will have height in your sample, then it is, you can see here. So here when you will see, this is like when your light is passing through a different, suppose this is your sample and your light is coming, so it is giving this kind of a I mean images, and when you will coincide this and or let's say what to say, when you will merge this, then this will give you a jet perception. Okay. So here it is same thing, it is saying that two identical bright field image is being overlaid slightly. So this is the two identical bright field images which is overlaid slightly offset from each other, and the subsequent interference due to the phase difference converting changes in phase. Okay, so that's why this is your light and this is your dark, which is here you can see. And because of this perception, because here two different, I mean images are overlaid with the slightly offset, it will give you the z axis, or you can say the depth perception of your image. So in the DIC only it is possible. So here you can see this is your 0% I think polarization, this is 90% polarization again. So here when your light will pass, so it will give a different polarized. Okay. So that's why when you will coincide or when you will merge these images, you will get this depth. So this is also, this is your depth perception in the DIC. So this, here you can clearly see Polson prism orientation is different. Okay. So the DIC image has the appearance of a three-dimensional object under very oblique illumination, causing strong light and dark shadows on the corresponding faces. The direction of apparent illumination is defined by the orientation of the Wolstein prism. Okay. So again, it depends the direction of your illumination. So here your Wolfson prism orientation is in this direction. It is in the this direction. So where you want to get your depth perception depends on the Wolfson prism. This interference may be either constructive or destructive, giving rise to the characteristic appearance of the three dimension. So it is more about this. So basically, in the DIC mode, you can see um, the depth perception of your images. So this is all about that. And yeah, so now we have covered some part of the this phase contrast, bright field, dark field, and DIC. Now we came to the polarized configuration or the polarized microscope. So the question from this microscope is that cross polarized configuration is shown in polarized light microscopy, DIC, phase contrast, or the DIC and polarized cross polarization. Do you know cross polarization, or uh, in which microscopy can you see the? Can you tell me? You want to see the question? cross polarized configuration is shown in which microscopy? Polarized DIC, phase contrast, or what? Or both? A polarized, A, A. Are you sure it is A? Let's see. So it is asking about the polarized, cross polarized. So it is our polarizer. So how we place? So firstly, this is our polarizer. Okay. So the first polarizer is placed beneath the specimen. So this is our specimen plane, and this is beneath the specimen. It means what? This is the first polarizer. This beneath the specimen. Another polarizer, which is nothing but the analyzer. We called it analyzer also. Is placed above the objective. This is your objective. This is above the objective. When both the polarizer and the analyzer are inserted into the optical path, their vibration azimuths are positioned at right angle. So this is at this horizontal. So this is at right angle at the 90 degree. So we place in such a way that it should make a position at right angles to each other. So this configuration, the polarizer and the analyzer are crossed with no light passing through the optical system and a dark background present in the eyepiece. So this is your cross polarized configuration. This is your one polarizer, and this is the both are at the right angle. Okay, these both are right angle. So the polarizer and the analyzer are crossed. So this configuration is called the cross polarized. Okay, so this is found in the DIC and the polarized microscopy both. So this is for the polarized, and in the DIC also your crossed polarizer is used. If you will see the lectures, I think I forgot the slide, but yeah, here also your cross polarizer, two polarizer is prism, two type of prism is used here also, Wolfson and the Nomansky prism. And also here your we use this polarizer. So cross polarizer techniques, this is your polar how it actually looks. So this is also used in case of your DIC. So in both DIC and in the this polarized light microscopy, we use cross polarization as shown in the question or in the figure. So in the both we use. Okay, cross polarization means simply you just understand here this is your polarizer 1 and this is analyzer, or let's say a polarizer 2. So here this is inserted in such a way that the this optical path between this are right angle to each other, or you can say are positioned in such a way that it will make a right angles or the 90-degree to each other. So this kind of a configuration is known as the cross polarization. Okay, so I hope is it clear that what does it mean by the cross polarization. Can you tell me the polarized light microscopy color images are formed due to the presence of what? Polarizer, analyzer, lambda plate, or both polarizer and the analyzer? So what is the answer for this? So Nishant is asking which type of sample can be used for this technique. So are you asking for the polarized light microscopy?
Yes, yes. And I just want to know that it is a reflected mode or a transmitted mode. Why the
What?
diagram is transmission, right? Whether we can use for the opaque material.
I mean
So,
for polarized.
yes, yes.
Okay, so polarized we can use it for the any kind of a metallurgical sample. Let's say a metallic sample also we can use for that, or other kind of a samples can also be used. I mean it can be used generic. I mean there is no any like in case of phase contrast we can use it for the biological, but for this I think we can use many of the samples and not any specific.
But here the light passes through the sample, no? That's how.
Where? Yeah, so here in this you are saying now. So any kind of a sample, it means to say the sample preparation is required so that light can be passed. It does not mean that, I mean, it depends on the sample preparation. So here, if you have any metallic sample, so for let's say, if you prepare a sample in such a way that it is very thin and light can pass, then it can be used for this microscopy also. I think. Let me just see. This is a one kind of a image of this. I think this is a metallic sample. So just so here it is about the lambda plate. So here you can see this is the, I think, images. Yeah, so this is a image from the polarized light microscopy. So we can use a metallic sample also here.
So all side of the sample should be polished.
Yeah, definitely. I mean, be- it is always advisable before using any kind of a because it should not have any surface roughness or uh, more impurity on the surface, so you can get a more feature of your sample.
Normally, when we do metrography, we do polish on one of the side, not every side.
Oh, yeah, yeah, definitely. You are asking about not every side, the surface or the portion which you want to see, that should be, you know, smooth. There won't be any, should not be any scratches, because if there will be scratches, then you can't be able to see the features of your substrate or what you want to see.
So what would be the thickness of the sample?
Thickness of the samples could be ranged. I have to see for this any particular ranges, because I have not used this microscopy. It is not available everywhere for some very specific labs and all. But let's say it could be, I I don't think so. I think it should be a very very thin in the range of micrometer. I mean, just like a hair thin.
Okay.
Yeah. So the answer for this question was the lambda plate. So here you can see the polarized light microscopy. Optical train also include an auxiliary element known as lambda, or the retardation plate, that is used for the quantitative analysis. In polarized light, the lambda plate converts the contrast into colors. Okay, so I mean, it will create a colors. So that's why you will get a lots of colors because of the different contrast of the different field. So as in phase contrast, optical path difference gives rise to color. Although this time with polarized light and the brief fringent material in the specimen, the path difference leads to the extension of certain wavelengths, because here the different in the paths therefore in the phase contrast you just see the not this kind of a variation in the color, but here you can see a different color. Okay, because here we are using a lambda plate, and that will convert the contrast into the color. Okay, so only certain colors remain from the white light and create beautiful color picture. So this is how, this is like a saying in this mechanism, that how you are creating this kind of a colorful images. And this is images, this is how you will get a image from when you will be using a polarized light microscopy. Okay, so you won't get a black and white kind of image like you get in any other samples, like in bright field or the dark field. Here you will get a colorful images because of the difference in the contrast of the different places. Yeah, so I have few more questions on this. So this is about the which optical microscopy works efficiently in identifying multi-phase material. So again, polarized, DIC, phase contrast, or the both. So here face identification by reflected. This is POM. POM is what? This is a polarized light microscopy. So polarized, here you can see a different, how you will, suppose if your material will having a different phase. So it is all, or let's say any metallic sample will have a, if you are see, if you want to see a different phase of your sample, okay, then it is good to use this kind of a microscope, where in this microscope offers an immense amount of information about the compositions. Okay, so different places can be having a different composition. Suppose if you are using a steel or the iron kind of a sample, then the different portion will have a different constituent of the element or the composition. and the three-dimensional structure of a range of the sample. Okay, so the valuable in manufacturing and research, polarizing microscope is relatively inexpensive and an accessible research and quality control tool that can provide information unavailable with any other technique. Okay, so here you can see this is your superconductor material. It constitute of your yttrium, barium, copper, and oxygen. So this is your tetragonal phase, and this is your orthorhombic phase with multiple twinning. Okay, so here you can easily see a different phase from this microscopy. So this kind of a material, just now somebody was asking that what kind of a, so this kind of a compound where you will be having a different constituent or the different composition in your compound or alloy. Let's say you have a composite, or you want to see, let's say you have a suppose you will have some kind of a steel which consists or different, or the aluminum alloy or any kind of alloy, and it consists of different compositions, then it is good to use this microscopy. So yeah. Now, the next is in fluorescence microscopy. Uh, Excitation and emission signals are differentiated by what? Excitation filter, emission filter, dichroic mirror, or the eyepiece. So yeah, this is the component when you will see the lecture of the fluorescence microscopy. So this is your sample. Here, this is your light source will go from here. This is your excitation filter. This is your dichroic mirror. So here it will go, and this is your objective. Here you will be having prism, and this is your eyepiece where you will be seeing, and this is your sample. Okay, so the specimen is illuminated with the light. So this is your sample, and the it will illuminated light will go from here, and which is absorbed by the fluorophores, causing them to emit light of longer wavelengths. Illumination light is separated from the much weaker emitted fluorescence through the use of spectral emission filter. Typical components in this microscope is light source. The first one here in the form of light source, we use xenon arc lamp or the mercury vapor lamp. This can be asked in the question that what kind of a light source can be used in the fluorescence microscopy. Excitation filter, dichroic mirror, or you can say the beam splitter, or emission filter. Emission filter is here. So because of the tremendously sensitive emission profiles, spatial resolution, and the high specificity of fluorescence investigation, the technique is an important tool in genetics of the genetics and the cell biology. So for the fluorescence, for the cell biology and all, we can use a fluorescence microscopy. So here the excitation and emission signals are differentiated by what? Dichroic mirror. Okay, so this will a beam splitter. So this is, it can differentiate, or it can between the excitation and the emission signal. Your, because this is your emission filter, from here it will come, this will go from the emission, and this is your excitation. So in between the excitation and the emission, you will be having a beam splitter or the dipole mirror. So I have few more questions, but let's discuss it in the next class. I think I have to close this class. So yeah, I hope some parts are clear for today's session. So the Arun is saying in the last assignment, last problem was about the calculation, the depth of field. Same for, yeah, I have shown you, I think in the last class, how to calculate the depth of field. Yeah, so if it is, no, I think it is given in the lecture also. You just see the lectures, whatever the assignments problem are given, they all are from the your lectures only. So there won't be any other problems apart from your lectures. So yeah, thank you guys. Let's meet in the next class.